Welcome back to the show, everyone. Hope you're doing well. Today we're talking about the Wuhan Lab Leak again, going into a little bit more detail. Today we're in the medical journal called Springer. If you just go to their About Me section, they provide millions of scientific documents in the form of journals, books, references, all for free. Springer is a member of what's called Springer Nature from Switzerland. It's an additional research repository for medical and scientific literature. And Springer from Switzerland, they're involved with the Nature magazine. It's all part of the same group from Switzerland here. So just a little background on this journal publication today. We're in the Journal of Environmental Chemistry Letters. It's a journal that interfaces geology, chemistry, physics, and biology, interdisciplinary. So yeah, today we're in the publication called Should We Discount the Laboratory Origin of COVID-19? We have a little bit of biographies for the contributing researchers. Rosanna from the Microbiology University in Austria. Eutherm Genetics in Canada. Deep Learning Solutions Institute in Japan, University of Santiago in Spain, Florida, LLC in Florida, University of Pittsburgh School of Medicine. So all these people are definitely legitimate researchers. Let's go into what they talk about today. There is a near consensus view that severe acute respiratory syndrome coronavirus, the causative agent of COVID-19, has a natural zoonotonic origin. However, several characteristics of the SARS-CoV-2 taken together are not easily explained by zootronic origin hypothesis. These include the low rate of evolution in the early phase of transmission, the lack of evidence for recombination events, a high pre-existing binding to human angiostensin converting enzyme ACE2, a novel furin cleavage site insert, a flat ganglioside binding domain, GBD, we've gone over that in the previous video, of the spike protein, which conflicts with the host evasion survival patterns exhibited by other coronaviruses. Very important aspect. And also a high human and mouse peptide mimicry, which is again very unusual. Initial assumptions against a laboratory origin, by contrast, have remained unsubstantiated. Furthermore, over a year after the initial outbreak in Wuhan, there is still no clear evidence of zoonotonic transfer from a bat or intermediate species. On the TV, they make it seem nice and cut and dry, that it's all Trump propaganda, but that's not the case. There are legitimate reasons why we should be looking into this as a people. It goes on to say SARS-CoV-2 is a natural beta coronavirus of lineage B, and the causative agent of COVID-19. The first detected cases were identified in Wuhan, December 2019. Bats are thought to be the natural viral reservoir and have been identified as the ancestral source from which the severe acute respiratory syndrome coronavirus evolved. While several intermediate host species have been proposed as the zoonotonic source of SARS-CoV-2, the source of direct bat to human or intermediate animal to human zoonotonic transmission has not yet been established. Over a year later, An alternative hypothesis that SARS-CoV leaked from a laboratory has been widely dismissed, yet very few papers counter this theory with actual data and analysis. So this is beyond what we're used to seeing on the TV. And now we're in essentially a situation where big pharma, companies like Pfizer and Moderna, are creating these so-called vaccine RNA gene therapies as if this is a naturally occurring virus. But there is ample evidence that has determined that this is not a naturally occurring virus. And in the previous video where we went over the Dr. Yan, researcher from the Chinese Institute of Virology, there are several reasons why this is the case, that the COVID-19 does appear to be a bioweapon genetically engineered for the reasons that we went over in the previous video in the Wuhan Institute of Virology.